much. And Master Protocol is an evolving thing in this country and also internationally. The overall goal is, of course, the question, how can we improve drug development? How can we faster identify effective drugs? How can we make the approval process shorter? And how can we together also identify companion diagnostics? So the master protocol in lung cancer, the concept was born when we had a meeting in 2012. I happened to be at that time in the steering committee for CTEP and was asked to, together with Shakun Malik at that time in FDA, to put together a meeting between industry, academia, NCI, and FDA. And the task was to discuss how can we integrate better biomarkers into clinical trial designs. So we had this meeting, and the meeting report is here, and the concept of master protocols was born. Now, on the same time we were discussing master protocols in this setting, the group Friends of Cancer Research had a task force also, and they worked also with FDA on how to develop master protocols, and Roy Herbst was chairing this Friends of Cancer task force. So two parallel organizations was working on the same concept, how to develop master protocols in lung cancer. So of course we had to find a way to work together, which has developed as a wonderful teamwork, which I will describe. First of all, what is master protocols about? We have two types, main types of master protocols. We have the umbrella trials, where we are focusing on one single cancer type. And we have within lung cancer, for example, we have a lot of abnormalities and link them to clinical trials. That is umbrella trials. And I will talk about Alchemist, I will talk about LungMap, but we have also basket trials, which means we don't care about tumor type. The only thing we care about is the molecular abnormalities and link it to clinical trials. And an example of that is the NCI match trial, which I will describe. Now, Alchemist has been described earlier in this meeting by several presenters, so I don't want to go in detail here. That is for early stage lung cancer, and the concept here is to screen a large number of patients. As a matter of fact, I think it is around 8,000 patients, which is planned to be screened by NGS technology for multiple mutations, and then connected to some clinical trials. Until recently, it has been only 2 clinical trials linked to those, one to EGFR mutation and one to ALK, but most recently there has been introduced a non-matched arm, which has been discussed also here, which is nivolumab versus placebo. So that was a good thing, because then you can offer patients who haven't matched abnormality also some treatment. LungMap is another setting. LungMap is squamous lung cancer, and it is advanced disease and second-line therapy. Now, LongMap is a unique private-public partnership including several organizations. It includes, first of all, the cooperative groups, Friends of Cancer, which I mentioned, Foundation for the National Institute of Health, NCI, and FDA are all sitting around the same table and are discussing trial design and new drugs. So what is the trial design? Trial design is we are starting out with a phase 2 trial with new drugs, and if the phase 2 trial achieve defined landmarks, it goes smoothly into a phase 3 registration trial. However, it is a possibility that if, of course, the phase 2 trial achieve so dramatic effect, you can eventually get an approval already there. But you need the registration trial. I don't want to go into the biostatistics. That has certainly taken a lot of effort to as a part of the design. But all the patients with squamous lung cancer, advanced disease, are undergoing genomic screening from Foundation Medicine's platform, Foundation One. In addition to specific biomarkers requested by some companies which goes beyond the mutation analysis. The primary interim endpoint is progression-free survival and also the overall survival, and it is the idea to randomize new investigational drugs with compare it to standard of care. This is the original design. We do the biomarker profiling. We have biomarker A, B, C, and D. And as I said, we randomize the experimental drug to standard of care. And we have also a non-matched arm, so we can offer all the patients some treatment. This is the original drug design and drug selection. We have a PI3 kinase inhibitor, CDK4/6 inhibitor, FGFR inhibitor, and a MET inhibitor originally, and in the non-matched arm, and PD-L1 inhibitor. Now, MET went out of the design based on disappointing results the company had, primarily in some other tumors, but also affecting lung cancer. So this arm went out, and we are working on other sub-studies, which I will come back to. I don't think I will go into details around the biostatistics here, but this is have certainly a certain predefined landmarks. Here is a sample size we are looking for in the phase 2 and in the phase 3 studies. But a lot of things happened since we designed the LongMap study. One of the major things happening in this space was, of course, approval of nivolumab in second-line squamous setting. So the LongMap team had to figure out how do we deal with that in this setting, and how can we move forward. So as a matter of fact, it showed up that the LongMap trial design is quite flexible. We originally talked about second line squamous treatment. Now we could add a third line and put docetaxel into the third line setting using nivolumab in the second line setting as a new standard of care. So there is some flexibility. The whole idea in this long map study is, of course, if a drug doesn't achieve the predefined landmarks, it can be replaced continuously with another drug. We have two non-matched sub-studies introduced, and we are in the process of getting that into practice. It is for nivolumab naive patients. We have an arm planned for nivolumab plus ipilimumab versus nivolumab as single agent. And for patients who are previously exposed to an PD-L1, PD-1 antibody, we have durvalumab and tremi combination. We have also negotiated with with BioMarine for another study. So this is the non-matched sub-studies and the matched sub-studies as we expect them to look like when everything is approved, and it looks like it will be. So long map and master protocols are not easy things to launch. There is a lot of components to it. There's a lot of organizations involved, and we have almost phone conferences 3 to 5 times a week in different committees to have this moving forward. We have an oversight committee, an executive committee, we have a drug selection committee who evaluate the whole time new drugs to be introduced in this setting. A lot of work has been put in by the study chairs, particularly Roy Herbst and Valley and David Gandara, but also the other people involved. Mary Redman is a crucial person for the biostatistics analysis. Okay, that was long map. Now we go to the basket trial, NCI match trial, and that is also, as I said, independent on tumor type, focusing on molecular abnormalities. And we can see here there is a lot of molecular abnormalities to choose from. And just a second. There you go. So NCI match trial is, they have several clinical trials linked to the abnormalities. And of course, it is a simple drug design. If you have stable disease or response, you continue. If you have progressive disease, you go out. But you have the opportunity, if you at progression also identify another molecular abnormalities, you can link to another trial. And finally, master protocol concept is emerging, and we are trying to find new areas where we can introduce master protocols. One of the areas which we seriously has considered is for ALK positive patients, and emphasize that the design is in progress. We have had a lot of negotiations around this. The idea here is to evaluate new drugs, but also to evaluate the sequencing of the new ALK inhibitors. What is the most optimal sequencing for use of those new drugs? So don't pay too much attention to the drugs and the trial design because we are still negotiating this design. So that is the master protocols here in this country I'm aware of. We have also been talking of a master protocols for small cell lung cancer. This has been preliminarily discussed. Again, I would like to invite you to a very exciting meeting in San Diego in January. Karen Kelly is one of the chairs, so I guess you agree that we should invite everyone to come there. Thank you.
So Fred, before you leave, thank you for all your efforts on these master protocols. I think tying back to my talk on financial toxicity, a great way to reduce financial toxicity for patients is enroll them in clinical trials, and particularly the lung MAP trial, because that's providing a free foundation medicine analysis and then free immune checkpoint inhibitor for most patients that might be spending thousands and thousands of dollars. So,
yeah.
So thank you.
Just really quick, because there's as we heard all these targets and everything, how quick? Because one of the things, like you said, is to speed things up. So how quick can a target and a drug potentially get put into those studies? Because I know when you start getting all the different groups that are in there, somebody comes up with a unique target. I would like to get this in. Here's my drug that targets it. How quick can that be implemented?
So it is difficult to give an exact timeline. I can tell you the mechanism. The drug selection committee in LongMap is working continuously with many companies for new targets, and as soon as we can see some encouraging, I mean, we don't want phase 1 studies to be included here, so we need to see some activity from the candidates. But as soon as we see that the infrastructure is already established, so it can go very fast to come with an agreement with the company for a new clinical trial. I don't know exactly what a fixed timeline for it, but it goes very, very efficient, and the drug selection committee is very active.
Thank you.